Assalamu alaikum. Good morning. Welcome to the last lecture of the digestive system physiology. Today we are going to cover the physiology of the small and large intestines. The objectives of today's lecture are to understand the motility patterns of the small intestine, to briefly describe the digestion and absorption of carbohydrates, proteins, lipids, electrolytes, water-soluble vitamins and vitamin B12 in the small intestine, and to describe the digestive and absorptive activities that occur in the large intestine which lead to fecal material formation, and to contrast and compare the colonic motor activity during a mass movement with that during hostration and the consequence of each type of colonic motility, and to describe the sequence of events occurring during defecation, differentiating those movements under voluntary control and those under intrinsic involuntary control. The small intestine is the major site for the digestion, both mechanical and chemical, and absorption of food, salts, water, and orally administered drugs. It serves to mix digestive secretions with food and propel chyme along the intestine from the pylorus to the ileocecal valve, digests and absorb nutrients, absorb the constituents of digestive secretion. It doesn't absorb only nutrients but also absorbs the digestive secretions after they perform their function. Also the intestine serves to regulate gastric function, and we saw examples on that in the control of gastric motility and gastric secretion, and mainly the effect of cholecystokinin and secretin, as well as the neural reflexes. It serves to synthesize and secrete hormones that influence the feeding pattern and the gastric function. Finally, it defends the body against invasion by microorganisms. As you studied in anatomy, the small intestine is composed mainly of three parts: the duodenum, the jejunum, and the ileum. And it extends between the stomach and the large intestine from the pyloric sphincter to the ileocecal valve. So the small intestine is the longest part of the alimentary canal. It has a length of 6 to 7 meters in cadaver or in the dead body due to the loss of muscle tone, and 2 to 4 meters during life. It has three subdivisions or three parts: the duodenum, which is the shortest part. It is mostly a retroperitoneal structure. It has a length of 25 centimeters. The jejunum, it is intraperitoneal, surrounded by the peritoneum or the visceral peritoneum, except at the attachment of the mesentery. It has a length of 2.5 meters. The ileum is the longest part of the small intestine. It has a length of 3.6 meters, and it is again an intraperitoneal structure. The small intestine is innervated by the parasympathetic division of the autonomic nervous system. The fibers run through the vagus nerve, and the sympathetic division of the autonomic nervous system through the thoracic splanchnic nerves that in turn modulate the enteric nervous system. So both divisions of the autonomic nervous system, parasympathetic and sympathetic, they modulate the activity of the myenteric and submucosal plexuses. The arterial supply is primarily from the superior mesenteric artery, and veins drain into the hepatic portal vein via the superior mesenteric vein. Histologically, The wall of the small intestine, like other parts of the alimentary canal, is formed of the 4 basic layers. These are from in to out: the mucosa, the submucosa, the muscularis, and the serosa. The wall of the small intestine is composed of the same 4 layers that make up most of the alimentary canal. The mucosa, that is composed of simple columnar epithelium that contains many types of cells. One type of cells are the absorptive cells or absorptive enterocytes. They digest and absorb nutrients. And again, I repeat, they can digest because they express digestive enzyme on their surface. So they will perform the last step of digestion before, or chemical digestion, before they absorb the nutrients. The other type of cells are the goblet cells. These are cells that produce mucus, which is important for lubrication and the protection of the mucosa from any damage. Other cells are also present in the mucosa. These are the cells that line the intestinal glands, and we will talk about them in the next slides. The mucosa also contains or is formed of the lamina propria, which is a loose connective tissue, and the muscularis mucosa, like the other parts of the alimentary canal. Next to the mucosa is the submucosa in the duodenum. The submucosa contains duodenal or Brunner's glands that secrete an alkaline mucus that helps neutralize gastric acid in the chyme. So it is very important, it has very important function because it is the first part of the duodenum that receives the acidic chyme. So it is very important, or the function of this gland is important in neutralizing the acidic chyme as it enters the duodenum from the stomach during gastric emptying. The muscularis externa is composed of 2 layers like the esophagus. There is no third layer or no internal oblique layer, and the usual serosa, which is part of the visceral peritoneum. Here we can see the mucosa. We have the villi or the finger-like projections of the mucosa that contain a core of lamina propria. And this core of lamina propria contains the lacteals or the lymphatics, plus the arteries and vein. And between the villi we can see that there are glands or crevices between the bases of the villi. These are called the intestinal glands, and also we call them the crypts of Lieberkühn, the scientist who first described them. In the villi are covered by the absorptive cells that absorb nutrients, and the apical membrane is projected in small finger-like projections. We call them the microvilli. Between the absorptive cells we can see the goblet cells that secrete mucus, which act as a lubricant and as a physical barrier. In the intestinal gland, or in the crevices of the crypts of Lieberkühn, we can find other types of cells. We can find the enteroendocrine cells that secrete hormones like secretin, cholecystokinin, and gastric inhibitory peptide. Also Paneth cells that secrete lysozyme, a bacteriolytic enzyme, and these cells are also capable of phagocytosis. Here in this figure, the third cell that is present in the intestinal gland is not shown. It is the secretory cell. These are cells that secrete the intestinal juice, and maybe I will mention it in the coming slide. So the small intestinal mucosa contains many deep crevices lined with glandular epithelium. Cells lining the crevices form the intestinal glands or the crypts of Lieberkühn and secrete the intestinal juice. The intestinal glands contain secretory cells, stem cells that give rise to all other types of cells. The Paneth cells that we saw them in the previous slide secrete lysozyme, bactericidal enzyme, and are capable of phagocytosis. And enteroendocrine cells, there are many enteroendocrine cells. The main three types are S cells that secrete the hormone secretin, CCK cells that secrete the hormone cholecystokinin, and the K cells that secrete the hormone gastric inhibitory polypeptide. Please be careful that these three hormones or the products of the enteroendocrine cells are released into the blood, and they are not part of the intestinal juice that will be released into the lumen of the small intestine. These cells form the exocrine part of the intestinal gland, while these three cells form the endocrine part of the intestinal glands. Since the small intestine is the longest part of the alimentary canal, and it is the main absorptive or the main site of digestion and absorption, so we need that the small intestine has a large surface area. The larger the surface area of the small intestinal mucosa means the more efficient absorption. So there are structural features of the small intestine that increase the surface area for absorption, and this will facilitate the process of absorption. One of the structural features is called the plica circularis. These are circular folds of the mucosa and submucosa that are found throughout the small intestine, and these circular folds will increase the surface area. Here we can see these plica circularis that are composed of mucosa and submucosa. Then the mucosa itself, it has finger-like projections, and we call these finger-like projections the villi. The villi are only composed of mucosa, so epithelium and lamina propria. If we take one of the surface absorptive cells, we can see that its apical membrane has very small microscopic finger-like projections, and those projections we call them the microvilli. So these three structural modifications will greatly increase the absorptive surface area in the small intestine. So again, here we can see the plica circularis, and the plica circularis is covered by mucosa. The mucosa has finger-like projections called the villi. This is magnification. We have here 5 villi, and this is one villus. If we take one cell here and we focus on that cell, we can see that the apical membrane, which is a phospholipid bilayer, is projected in very small finger-like projections that we call them the microvilli. So larger mucosal surface area means more efficient intestinal absorption. These structural features that increase the absorptive surface area are the plica circularis, and as I said, these are circular folds of the mucosa and submucosa forming permanent ridges, and each ridge is about 1 centimeter in height. The other structural modification that contributes to an increase in the surface area are the villi. These are finger-like projections of the mucosa. They have a height of 0.5 to 1 millimeter. Each villus is covered by epithelium and has a core of lamina propria that contains blood vessels and lymphatics. The lymphatics are called lacteals. In 1 square millimeter, we can find 20 to 40 villi. So if we take 1 square millimeter of the intestinal mucosa, we can find up to 40 villi. This will vastly increase the surface area of the intestine. The microvilli, projections of the apical or free border of the membrane or the free membrane of the absorptive cells. The microvilli are about 1 micrometer in height, and they, as I said, they are part of the apical membrane of the absorptive cells. If I take 1 square millimeter of the intestinal mucosa, I can find 200 million microvilli. Again, this large number of microvilli will further increase the surface area. These structural modifications will increase the total surface area of the mucosa. It increases that surface area 600 times. So if we don't have these structural modifications, the surface area of the small intestinal mucosa is only 0.33, 1/3 of square meter. But because we have these structural modifications, the actual absorptive area of the small intestinal mucosa is 200 square meter, which is very good for very efficient absorption of nutrients. Now let's talk about the intestinal juice and the brush border enzymes. The intestinal juice is clear yellow fluid. The small intestinal glands produce around 1 to 2 liters of juice per day. It contains water, mucus, and is slightly alkaline because it contains bicarbonate. The pH is 7.6. Together, pancreatic and intestinal juices provide liquid medium that aids the absorption of substances from chyme and the small intestine. Please notice that intestinal juice doesn't contain any enzymes. Before I start to explain the brush border enzymes, as you see in the intestinal juice composition, it contains water, mucus, and ions, but no enzymes. 0 enzymes are present in the intestinal juice. So the intestinal glands do not produce digestive enzymes. The enzymes in the small intestine are integral membrane proteins that are present in the microvilli, in the apical membrane of the absorptive cells. And since they are present in the microvilli that look like brush border, we call them the brush border enzymes. Brush border enzymes are digestive enzymes synthesized by the intestinal absorptive cells and inserted in the plasma membrane of the microvilli. That's why we call them the brush border enzymes. Many enzymes, or we have different types of brush border enzymes, and these include 4 carbohydrate digesting enzymes. These are dextrinase that digest dextrine, maltase, saccharase, and lactase. Protein digesting enzymes called peptidases, like aminopeptidase and dipeptidase. If you remember, the pancreatic juice contains carboxypeptidase. As you know, the polypeptide chain, it has an amino terminal and a carboxy terminal. The carboxypeptidase will clip off amino acids from the polypeptide chain from the carboxy terminal. The aminopeptidase will digest the polypeptide chain starting from the amino terminal. That's why we call it aminopeptidase. The digestion of polypeptides will also produce dipeptides, 2 amino acids joined together. So these dipeptides are digested or split by the enzyme dipeptidase, which is a brush border enzyme. There are also 2 nucleotide digesting enzymes. These are nucleosidases and phosphatases. What are the features of the small intestinal motility? The small intestinal motility, we have 2 different patterns, and it depends whether we are talking about the motility pattern during the fed state or during the meal, or the motility pattern during the interdigestive period or between meals. So in the digestive periods or during meal, we have two motility patterns: segmentation, which is mainly a mixing contraction, and peristalsis, which is a propulsive movement. So segmentation is mainly meant for mixing contents inside the lumen of the small intestine. Peristalsis is mainly meant to propel or push the chyme towards the ileocecal valve. During the interdigestive periods, we have one motility pattern called the migrating motility complex or migratory motility complex, which is MMCs. Let's take these motility patterns in more details one by one. Segmentation, we said it is mainly for mixing. It is the small intestine's primary motility pattern during digestion or during meal. So it is the main. If I ask you what is the main motility pattern of the small intestine during digestive periods, it is segmentation. So it is the small intestine's primary motility during digestion of a meal. It mixes. Its main function is mixture, but also it propels slowly the chyme. And we need that slow propulsion of chyme down the small intestine because we need to provide longer time for chyme to stay in the small intestine, so it will be properly digested and adequately absorbed. The segmentation consists of oscillating ring-like contractions of the circular smooth muscles of the muscularis externa along the small intestine's length. So between the contracted segments are relaxed area, as we see here in this figure. So we have contractions at regular intervals, and between the contractions we have relaxed areas. So what will happen next? The contractile rings here will relax and the relaxed rings will contract. So this will divide the chyme into different segments, and this will ensure better mixing of the chyme. So the contractile rings occur at regular intervals every few centimeters, dividing the small intestine into segments like a chain of sausages. These contractile rings do not sweep along the length of the intestine as peristaltic waves do. So these contractions don't move, so just they contract and then relax. No contraction will sweep down the small intestine like what happens in peristalsis. After a brief period, the contracted segments will relax and ring-like contractions will appear in the previously relaxed areas, and this will ensure mixing of the chyme with the intestinal and pancreatic secretions, as well as with bile. Peristalsis, which is the other motility pattern that happens during digestion of meals. In addition to the propulsive effect of segmentation, chyme is also propelled through the small intestine by peristaltic waves. These can occur in any part of the small intestine, and they move towards the anus or towards the ileocecal valve at a low velocity, which is 0.5 to 2 centimeters per second. These peristaltic waves are faster in the proximal intestine and slower in the terminal part of the small intestine. They are normally weak and usually die out after traveling only 3 to 5 centimeters. So this forward movement of the chyme is very slow. So the combined effect of segmentation and peristalsis will move the chyme in the small intestine at an average speed of 1 centimeter per minute. And as I said, this slow movement is required because this will give time for the proper digestion of the chyme and the absorption of the nutrients. This means that we need 3 to 5 hours to pass the chyme from the pylorus to the ileocecal valve. So this slow propulsion of chyme will make the intestinal transit time or the intestinal emptying time about 3 to 5 hours. Now we talk about the main motility pattern that happens during the interdigestive periods or between meals. It is the migrating or migratory motility complex. During periods of short fasting, when most of the meal has been absorbed, the stomach and small intestine exhibit a unique motor activity. Digestion and absorption doesn't mean that all the food or all the chyme is absorbed. So what is left over should be removed from the small intestine, from the stomach and small intestine, to clean the stomach and small intestine in preparation for the next meal. So this motility pattern is to remove any remnants from the previous meal. Intestinal segmentation contractions will cease between meal, and they will be replaced by the migrating or migratory motility complexes, or we call them the intestinal housekeeper activity, because they will clean the small intestine in preparation for the next meal. Migrating motility complex cycles that repeat every 100 minutes will sweep the intestine clean between meals. MMCs are regulated between meals by the hormone motility, which is secreted during the unfed state or between meals by the endocrine cells, the M cells that are present in the small intestinal mucosa. So they are one of the enteroendocrine cells present in the intestinal glands. When the next meal arrives, the migrating motility complexes will cease and the motor activity associated with the meal will take over. So motiline release will be inhibited by the meal, and now the motility pattern will become segmentation and peristalsis. What are the factors that regulate motility of the small intestine? Segmentation is stimulated by distension. So whenever any segment of the small intestine is distended by chyme, segmentation will result. Parasympathetic innervation will increase motility. As we know, parasympathetic system is stimulatory in the digestive system. Sympathetic innervation decreases motility. The chemical digestion in the small intestine, digestion of carbohydrates. The following enzymes digest carbohydrates in the small intestine. Pancreatic amylase will digest starch, glycogen, and the products of starch digestion that were produced by the action of salivary amylase like maltose, maltotriose, and alpha-dextrins. Alpha-dextrinase, which is a brush border enzyme, will clip off glucose from alpha-dextrins. Glucose is a monosaccharide that will be easily absorbed. Saccharase, brush border enzyme, will break saccharose into glucose and fructose. Both are monosaccharides that can be easily absorbed. Lactase, brush border enzyme, digests lactose into glucose and galactose. Both are monosaccharides that will be absorbed. Then maltase, which is a brush border enzyme, will split maltose and maltotriose into 2 or 3 molecules of glucose that will be eventually absorbed. The pancreatic amylase has no effect on other polysaccharide called cellulose. Plants or the fiber of plant or cellulose is not digested by pancreatic amylase. So it is an indigestible plant constituent as it moves through the digestive system, and it will be removed from the body with the fecal material. So here we can see a summary of the chemical digestion that happens in the small intestine, and where the enzymes on the luminal surface of the small intestinal cells, or the brush border enzyme, will digest the poly and disaccharides into monosaccharides that can be absorbed easily. Digestion of proteins. The following enzymes digest proteins in the small intestine. Pancreatic enzymes digest proteins into peptides like trypsin, chymotrypsin, carboxypeptidase, and elastase. These enzymes were explained in the previous lecture. The brush border peptidases digest peptides into single amino acids like aminopeptidase and dipeptidase. Digestion of lipids. Most of the lipid digestion occurs in the small intestine by pancreatic lipase. Some digestion occurs by lingual and gastric lipase, but this digestion is very minimal or minor. The lipase will break down triglycerides into free fatty acids and monoglycerides. And here we see a summary of the chemical digestion that happens in the small intestine. Carbohydrates will be digested finally to monosaccharides, proteins into the simple molecules, amino acids, and lipids into fatty acids and monoglycerides. Nucleic acids will be digested into pentoses, phosphates, and nitrogenous bases. Here we can see a summary of the absorption of all the nutrients like glucose, galactose, fructose, amino acids, and the amino acids, the short chain fatty acids, and the large short chain fatty acids, the long chain fatty acids, and the monoglycerides. As you notice, the fatty acids and monoglycerides will move or enter the enterocytes by simple diffusion. They will assemble to form chylomicrons that will leave the cell by exocytosis, and they will enter the lacteals, the lymphatics that are present in the villi. The other nutrients are usually transported by active transport or by facilitated diffusion, and finally they will enter the blood or the blood vessels in the villi. So absorption of carbohydrates happens by facilitated diffusion or active transport. Proteins are absorbed by active transport, lipids by simple diffusion, monosaccharides, amino acids, and short chain fatty acids will enter the blood, will pass into the blood. Long chain fatty acids and monoglycerides will be surrounded by bile salts forming tiny spheres called micelles. They will approach the brush border of the absorptive cells. Then the long chain fatty acids and monoglycerides will diffuse out of the micelle into the absorptive cell. So the micelle will act as transport vehicle from the lumen of the small intestine to the brush border. Inside the smooth endoplasmic reticulum of the absorptive cells, triglycerides are reformed and packed into chylomicrons. These are large globules with phospholipids, cholesterol, and proteins that leave the absorptive cells by exocytosis, and they will enter the circulation via the lymphatics or the lactea. This figure I already explained in the previous lecture how the lipase will act on the emulsified fat droplets to release monoglycerides and free fatty acids that will enter the micelle. Micelle will approach the brush border and will carry or deliver those digestion products to the absorptive cells. Inside the absorptive cells, triglycerides are reformed, packed as chylomicrons with the help of smooth endoplasmic reticulum. Chylomicrons will be released by exocytosis, and will enter the lacteals and will go back to the circulation. Emulsification is the process by which fat globules are broken into smaller pieces by the detergent action of bile salts, and I already explained that in the previous lecture. Here we can see how the different nutrients, or most of the nutrients, go back to the circulation through the blood, except for the chylomicrons that will enter the lacteals or the lymphatics and will go back to the circulation via the thoracic duct. With this, we finish the small intestine. Then we start with the large intestine. The large intestine is composed of several parts, as you studied in the anatomy. The large intestine is the terminal portion of the GIT that extends from the ileum at the ileocecal valve to the anus. It has a length of about 1.5 meters. Structurally, the four major regions of the large intestine are the cecum, the colon, the rectum, and the anal canal. The colon is composed of ascending colon, transverse colon, descending colon, and the sigmoid colon. The overall functions of the large intestine are the completion of absorption, the production of certain vitamins, especially vitamin K, the formation of feces, and the expulsion of feces from the body by the process of defecation or the defecation reflex. Again, the wall of the large intestine has the same four layers: the mucosa, submucosa, muscularis, and serosa. The wall of the large intestine contains the typical four layers found in the rest of the GIT. The mucosa, composed of simple columnar epithelium that contains absorptive cells, function primarily in water absorption, and goblet cells that secrete mucus for lubrication. Again, lamina propria, muscularis mucosa. Both absorptive and goblet cells are located in long, straight, tubular intestinal glands. So the large intestine mucosa also contains intestinal glands or crypts of Lieberkühn, but the cells inside or lining the crypts of Lieberkühn are completely different from the cells that are present in the crypts of Lieberkühn of the small intestine. In the small intestine, we find this mainly the secretory cells that produce the intestinal juice. The crypts of Lieberkühn in the large intestine, they contain absorptive cells and goblet cells. So, similar to the cells on the surface. The other layers are the submucosa, muscularis, and serosa. Here we can see a drawing of the small intestinal large intestinal mucosa with large intestinal gland or crypt of Lieberkühn. As you can see, this is the mucosa lining the large intestine with the opening for the intestinal glands. As you can see, the large intestinal glands, they contain absorptive cells that absorb water and goblet cells like the cells present on the surface mucosa. So there are no secretory cells in the large intestinal glands. What are the motility patterns that are seen in the large intestine? Very important to know that the large intestinal motility is not linked to the feeding process. So we don't see certain motility patterns that appear during a meal and other motility patterns that occur during the interdigestive state. So the motility pattern of the large intestine, unlike the motility patterns of the stomach and the small intestine, has nothing to do with the meal. So these are the same during and between meals. So the main motility patterns is hostration, which is similar to the segmentation that happens in the small intestine. These are slow segmenting movements that move the contents of the colon. They occur every half an hour or every 30 minutes. They are controlled by intrinsic plexuses or short local reflexes. And like in the small intestine, hostrations are stimulated by distension. Movements are slow, and these slow movements will allow the absorption of water and also will give chance for storing the fecal material until the conditions are suitable. So movements are slow, leading to absorptive and storage functions. The other motility pattern of the large intestine are the mass movements. These are propulsive movements. Usually hostrations occur in the proximal part of the large intestine, while mass movements occur in the distal part of the large intestine. These propulsive movements are increased motility in the ascending and transverse colon occur after a meal. This will drive the colonic contents to the distal portion of the large intestine, and the mass movements are triggered by the gastrocolic and duodenocolic reflexes. The gastrocolic reflex means that distension of the stomach will stimulate contraction of the colon or mass movement in the colon. Duodenocolic reflex again means distension of the duodenum with chyme will stimulate mass movement in the colon. And these are mediated by reflexes that run through the enteric nervous system, and we are talking mainly about the myenteric plexus. Also, mass movements are triggered by mucosal irritation. Any toxin or any microbe that irritates the mucosa can stimulate mass movement, and usually this leads to diarrhea. Again, mass movements are stimulated or triggered by intense parasympathetic stimulation, and we know that parasympathetic division of the autonomic nervous system is stimulatory in the digestive system. Now, what is defecation? Defecation is the reflex that expels the fecal material outside the body through the anus. When mass peristaltic movements push fecal material from the sigmoid colon into the rectum, distension of the rectal wall is the trigger for defecation. So usually between defecations, the rectum is not expanded or not distended. Whenever the rectum is distended with fecal material, mechanoreceptors in the wall of the rectum will get stimulated, and this will initiate the defecation reflex that will empty the rectum. As you can see here, the rectum, it is rich, or its wall is rich in mechanoreceptors or stretch receptors. If mass movement pushes fecal material into the rectum, distension of the rectum will stimulate the mechanoreceptors, and this will trigger the defecation reflex. Distension of the rectal wall will stimulate the stretch receptors in the wall of the rectum, and this will send action potentials through the sensory nerve fibers to the sacral spinal cord. And at the level of the spinal cord, the information will be processed, and motor impulses will pass through the parasympathetic nerves to the descending colon, sigmoid colon, rectum, and anus. So all of them will contract. There will be contraction of the longitudinal rectal muscles. This will shorten the rectum. The rectum will become shorter, so this will increase the pressure on the fecal material. Contraction of the diaphragm and abdominal muscles also will contribute to the pressure increase. This will open, or this high pressure will push the fecal material against the internal anal sphincter, so the internal anal sphincter will open. As you know, the external inner sphincter is under voluntary control. So if the conditions are suitable for defecation, the sphincter will be voluntarily relaxed, defecation will occur, and the fecal material will be expelled through the anus. If the sphincter is voluntarily constricted or the conditions are not suitable, defecation will be postponed, and the fecal material that is present in the rectum and causing distension and stimulation or activation of the defecation reflex will be pushed back into the sigmoid colon by something called retroperistalsis, backward movement of the fecal material from the rectum into the sigmoid colon to relieve the stretch or to reduce the stretch of on the wall of the rectum. If defecation doesn't occur, the feces back up into the sigmoid colon until the next wave of mass peristalsis will push this fecal material back into the rectum. What is the composition of feces? Majority of fecal material, though it seems to be solid or semi-solid, it contains water. 75% of the fecal material is water and 25% is solid matter. The pH of fecal material is slightly alkaline, between 7 and 7.5. The solid matter that is present in the fecal material, 30% of the solid matter is dead bacteria, 10 to 20% fat, 10 to 20% inorganic matter, 2 to 3% protein, 30% undigested roughage from the food and dried constituents of the digestive juices that were not absorbed, such as bile pigments and sloughed epithelial cells. The mucosal epithelial cells that were replaced by newer cells will be sloughed and will be part of the fecal material. The brown color of fecal material is caused by stercobilin and urobilin. Both are derivatives of hemoglobin degradation or bilirubin. The odor is caused principally by products of bacterial action, the action of bacteria on the fecal material inside the lumen of the large intestine, and these chemicals are indoles, scatols, mercaptans, and hydrogen sulfide. Let's quickly talk about the bacterial action in the colon. Numerous bacteria, especially bacilli, are present normally in the large intestine, especially in the absorbing colon, in the proximal part of the colon. This bacterial population of the large intestine is called the intestinal flora or the gut microbiota. The total number of bacteria in the intestinal flora is about 300 trillions, which is about 10 times the total number of cells in one human body. So we have more flora than our own body cells. They are capable of digesting small amounts of cellulose, providing few calories for their own metabolic use. So they depend on the digestion of cellulose to obtain energy and survive. The gut microbiota or the intestinal flora are harmless, and in fact they provide beneficial function. They are very important for us. They synthesize vitamins, vitamin K, B12, thiamine, and riboflavin, so they have nutritional contribution. Our daily vitamin K intake is not, or our daily dietary intake of vitamin K is not enough for or doesn't supply our requirements, so the missing amount is supplied by the gut microbiota. because they produce vitamin K that will be absorbed in the large intestine to be used in the production of certain, as a cofactor in the production of certain clotting factors. The bacteria formed vitamin K is especially important because the amount of this vitamin in the daily ingested food is normally insufficient to maintain adequate blood coagulation. As I said, again, the bacterial flora are, or the gut microbiota are beneficial because they raise the colonic acidity by the acid produced during the fermentation process. So this local acidity will promote the absorption of cations, the divalent cations like calcium, magnesium, and zinc. Also, bacterial gut microbiota enhance intestinal immunity by competing with potentially pathogenic microbes for nutrients and space. Colonic bacteria also produce various gases during fermentation like carbon dioxide, hydrogen gas, and methane. Most of these gases are absorbed through the mucosa and the remainder is expelled from the body as fletus or as gases. With this slide, we end today's lecture, and this is the last lecture of the digestive system. Thank you very much. Please, if you have any question, send me an email so I can reply to your questions and queries. Thank you.